Hello, my name is Nigel Mouncy. I am the director of the Joint Genome Institute in Berkeley, California. I was still an undergraduate at the University of Glasgow in Scotland on November 9th, 1989, and I remember seeing the TV coverage, but didn't fully appreciate how world-changing this event was at that time. Well, my father was a microbiologist, and we always had scientific journals lying around the house. Now, I remember thumbing through these, even when the words meant nothing to me, but looking at the figures and images and just being super curious. So my father never forced me to take the path I've taken to be a microbiologist. Naturally, it's what I've always wanted to do. Imagine if we understood the intricacies of life on our planet so that we would be able to harness nature to solve our problems in feeding, fueling, sheltering, and sustaining our world for many generations to come. What is the challenge your project addresses, and what is your solution? Over the eons, nature has presented us with a wonderfully interactive tapestry of organisms that produce polymers like cellulose, the most abundant on the planet, and the enzyme catalysts to produce them and to break down these molecules, and still others to transform them into products that could be made more sustainably than current and traditional methods. This is where the JGI comes in, as the only dedicated, collaborative public research resource focusing on characterizing the genomes of microbes, fungi, and plants. JGI's team has sequenced more than a petabase, that is 1,000 trillion letters of DNA code from thousands of bacteria, fungi, and plants from all over the planet, and made that information widely accessible for the public good through our data portals. Inside an abandoned mine in the far northern reaches of California, one of our collaborators scraped a pink biofilm growing in a hostile stream of acid waters and provided that sample to the JGI. Working together, we were able to identify the microbial players in this extreme environment, and later their DNA sequence signatures revealed a bacterial defense mechanism that we know today as CRISPR. This system has been used to create a suite of powerful genome editing tools, which is revolutionizing our ability to gain biological understanding in an unprecedented speed and scale that can help us, for instance, tailor plants to tolerate conditions such as drought and diseases that will proliferate as the climate continues to warm, providing hope for more robust food and fuel security. What microbes promise is billions of years of advanced intelligence that we are now just beginning to be able to decipher through the processes of DNA sequencing, functional exploration, and custom analysis with the power of supercomputing. Being able to take this knowledge and use it in ways such as producing renewable fuel, sustainable food, alternative energy, and new medicines that combat disease will sustain our planet for many future generations to come. If we just consider microbes, bacteria, fungi, and archaea, the branch of the tree of life living at the extremes of conditions, there are more microbes in just a handful of soil than stars in the Milky Way. And we know very little of who they are, what they do, and how they interact with each other and the environment they live in. However, we know that they are essential to life on this planet, including our own bodies. We have so much to learn to understand this complexity. The sheer magnitude of developing the detailed understanding of this rich tapestry of life requires us to continually think of new ways to measure, analyze, interrogate, integrate, and interpret relevant data at a tremendous scale. So I'm always thinking about the best ways in which we can develop capabilities and expertise to do this, whether that be new robotics that allow us to explore biological systems, or the development of computational tools that allow us to generate models to predict biology. Yes, these are very big challenges, but also wonderful opportunities. My father was a microbiologist. He worked on drug discovery, so he's always been very scientifically interested in the work that I've done throughout my career, and I think very proud of the fact that I inherited his microbiology genes and for the impact and importance of the work that I do today.